Now I come on to the single membrane organelle. Now we study about the single membrane organelle. That is these are follows. At first I come on to ER. First I come on to the ER, means endoplasmic reticulum. Endoplasmic reticulum, first the position of endoplasmic reticulum. If we observe a cell, then it is the cell membrane. It is the cell membrane, and in the center it is the nucleus. It is the nucleus. Now we can see that some tubular structure present or distributed from the nuclear membrane to the plasma membrane. Nuclear membrane to the plasma membrane in different side of the nucleus. Means it separates the chamber of the cell. It separates the cell into many chambers. Okay, so it separates the all chemical reaction which occurs inside a cell. Now I come on the component of the ear. If we observe the ear, we can see three components are participated to form the endoplasmic reticulum. These are cisternae. These are the cisternae, then tubule and vesicle. Okay, cisternae. Cisternae is a tubular structure. Cisternae is a plate-like structure. Cisternae is the plate-like structure which are parallelly arranged. Okay, and on the membrane of the cisternae, ribosome will present in some cases. Ribosome is present in some cases. Okay, then tubule. Tubule is a reticular structure which are branch tubular like structure which makes a network inside the cell. And vesicle is a spherical sac-like structure. And those cisternae, tubule and vesicle are togetherly form the ear, means endoplasmic reticulum. Then we come on the types of ear. In eukaryotic cell, their ear also divided into two groups, that is RER, rough endoplasmic reticulum, and ACR, smooth endoplasmic reticulum. RER means if the cisternae of the endoplasmic reticulum, contains the ribosome on the surface. For that the surface of the ear become rough. For that these endoplasmic reticulum are called rough endoplasmic reticulum. Okay, so because of for the presence of ribosome on the surface, this RER helps in protein synthesis and protein distribution. Protein synthesis and protein distribution inside a cell. Then we come on to the ACR. Smooth endoplasmic reticulum means if the cisternae of an endoplasmic reticulum does not contain any ribosome. For that, the surface of the endoplasmic reticulum will smooth. For that, these are called ACR, smooth endoplasmic reticulum. For the absence of ribosomes, they does not produces the protein, okay, or they does not helps in the protein synthesis, but they helps in fat metabolism, fat or lipid metabolism, or glycogen synthesis, or glycogen synthesis. It is the main function of the ACR. Okay, then we come to the next single membrane organelle, that is Golgi body. Okay, Golgi body is present near to the ear. It look like that. The main function of Golgi body is secretion. It present only in the plant cell. It present only in the animal cells. Okay, if we observe the structure of the Golgi body, we can see it has three components. These are the cisternae, microvesicle, and vacuole. These are the Golgi body composed of cisternae, microvesicle, and vacuole. Now we come to the structure of a Golgi body. Golgi body has two surfaces. One is the concave surface. And these are the cisternae arranged in a proper manner. Okay, it has two faces. We can see in the structure it has two faces. One face is concave face and another is convex face. The main function of Golgi body is secretion, and this cisternae of the Golgi body has some secretory part. For that, they secrete some substance which are carried to the convex part. For that, the concave face of the Golgi body, also called cis phase, and the convex phase is called trans phase. Okay, or it also known as maturation phase. Maturation phase, and this is also known as formation phase. Means the upper portion of the Golgi body secretes the substances which are put inside the put on the convex phase or trans phase. Okay, sorry, cis phase, not cis phase and trans phase. Okay, then these are the cisternae. Okay, then we come on to the microvesicle. Where does the microvesicle present? Microvesicle is formed in the transphase of the Golgi body. It is the immature sac-like structure. These are the immature sac-like structure called microvesicle. Inside the microvesicle, the secretory substances are put. Okay, the secretory substances are put on the in the microvesicle. Then, when the microvesicles are filled with the secretory substances, they become large and released from the Golgi body. This structure is called vacuole. Okay, when the microvesicle is filled with the secretory substance and become mature, then they are called vacuoles. Complete. Then another single membrane organelle, that is lysosome. That is lysosome. Lysosome is a spherical shaped single membrane bounded organelle which are formed from the Golgi body. Which are formed from the Golgi body. For that, lysosome contains the secretory substances which are produced by the Golgi body. And when the lysosome became independent, it contains more than 100 hydrolytic enzymes. The lysosome filled with the hydrolytic enzymes. Lysosome also observed in animal cell only. Plant cell does not have any lysosome and Golgi body also. In some cases, a special structure called as dictyosome, which are known as the Golgi body of plant cell. Why? Because it play the role of a like Golgi body. Okay, in plant cell. For that, dictyosome also known as the GB of plant cells. Same way, plant cells also has special single membrane boundary structures like lysosome is also called spherosome. Spherosome is the similar as lysosome, but spherosome present inside the plant cell, not in animal cell, but lysosome present in animal cell only. Okay. We can classify the lysosome as follows: that lysosome, primary lysosome and the secondary lysosome. What is primary lysosome? Newly formed lysosome are called primary lysosome. Okay, which does not take part in cellular digestion. Normally, the function of the lysosome is it helps in cellular digestion, but primary lysosome does not take part in cellular digestion. Then we come to the secondary lysosome. What is secondary lysosome? If the lysosome attach with another cell organelle or another part of a cell, if the primary lysosome attach with the another part of the cell, then the lysosome is called secondary lysosome. Secondary lysosome also classified into 3 groups. That is, these are autophagosome, heterophagosome, and residual body. If any lysosome attaches with the cell organelle, primary lysosome plus any cell organelle, any cell organelle is called any cell organelle is called autophagosome. Then we come on to the heterophagosome. If the primary lysosome attach with the food vacuole, then it is called heterophagosome. Food vacuole means the contractile vacuole, okay, which are formed at the time of phagocytosis. So primary lysosome when attached with the phagocytotic cavity or phagocytotic vacuole, then these are called heterophagosome. Then we come to the residual body. What is residual body? Residual body, if the primary lysosome attaches with the vacuole which contain the undigested food part. When primary lysosome attaches with the vacuole which contains the undigested food part. So it is undigested food vacuole, then it is called residual body. Remember it, the lysosome also called the pleomorphic organelle because they does not have any proper state. Commonly we can found in a cell the secondary lysosome. Which are attached with the another part of the cell, such as cell organelle, food vacuole or undigested food vacuole. Lysosome also called the suicidal bag. Why? Because it has many hydrolytic enzymes. Lysosome contains many hydrolytic enzymes. If any cell become diet or non-functional, then the lysosome of these cells all are burst. For that, all hydrolytic enzymes are put inside the cell and total cell completely digested. For that, the lysosome called suicidal bag because if any normal cell, if in a normal cell the lysosomes are burst, then the functional cell also be destroyed. For that, lysosome also known as suicidal bag. Now I come on to the next single membrane bounded part, that called vacuole. Vacuoles are the open space which are present inside the cells. If we can compare the plant cell with animal cell in respect to vacuole, we can see the animal cell has many vacuoles. But they are smaller in size. But inside the plant cell, the small vacuoles are fused together. All vacuoles are fused together and make a large vacuole. Okay, makes a large vacuole. For that, the nucleus moves from the central to the periphery side of the cell. This is called primordial utricle. This strait are called primordial utricle. Okay, then what is the primary utricle? Primordial utricle is the distribution of protoplasm for the vacuole, because the density of the protoplasm is higher in the periphery side in a plant cell for the presence of a central large vacuole. in a plant cell. For that, the distribution of the protoplasm of a plant cell is also known as primordial utricle. Each cell vacuole has a membrane is called tonoplast. The membrane of cell vacuole is called tonoplast. Tonoplast is the membrane. Okay. Then the vacuole. What does vacuole contains? Vacuoles contains the cell sap. Vacuoles contains the cell sap. For that, they regulate the osmotic pressure or osmotic potential of a cell. Sometimes the vacuole also contains the food matter, and sometimes they also contains the excretory products. Okay. Next, we come on to the third, membraneless organelle. Membraneless organelle. Membraneless organelle. First, centrosome. First is the centrosome. Where does the centrosome present? Centrosome only occurs in the animal cell. Okay. It is a star-shaped. Organelle which is present near to the nucleus. If we observe the structure of a centrosome, we can see centrosome has two barrel-shaped part which are present in 90-degree angle. Okay, look like that. Two barrel-shaped part. These barrel-shaped part are called centriole. And these centrioles are surrounded by astral ray. Like that. For that, they appeared as the star-like structure. It completely surround the centriole. Okay. Then this structure are called astral ray, which surrounds the centriole. If we come, then we come to the structure of the centriole. The structure of centriole is also known as cartwheel model. If we observe the structure of a centriole, then we can see it has a central hub. It has a central hub and bundles of 9 tritubular structure which surrounds the central hub. One, two, 1, 2, 3, Tritubular structure means 1, 2 and 3. 1, 2, 3. Another 9 tritubular structure. Each tubules contains each tubule contains 13 tubular protein. If we can observe its structure, these are called from inner side the A, B, C, A, B, C tubule. Okay, and A, B, C. Each tubule contains 13 tubular protein. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and 12 and 13. Contains 13 tubular proteins. Okay, each tubule. Then each tritubular structure interconnects through A with C, A with C. A with C. Okay, then some protein also present in the layer. The layer. Okay, and make a circle and make a circle, and 9 spokes is present there which connects with the central hub, which connects the 3, 4, 5, 6, 7, 8 and 8 and 9. So it has a central hub. It has 9 spokes, tritubular structure, central hub, spokes and tritubular structure. For that, this model also called cartwheel model. Okay, look like this. Then what is the function of the centrosome? Centrosome helps in cell division. Centrosome helps in cell division by forming spindle fiber in animal cell. If in the case of plant cells, their centrosome does not present. For that, In the plant cell, the spindle fiber will formed from the microtubule. Is formed from the microtubule. Microtubule means many tubular protein are arranged or make some tubules which are present inside the plant cells. They form the spindle fiber at the time of cell division of a plant cell. Okay. Then we come to the next membraneless organelle, that is very important, that is ribosome. That is ribosome. Okay. Ribosome is present in all types of cells, means prokaryotic, prokaryotic cells, and also in eukaryotic cells. Present in both plant and animal cells also. Okay. Ribosome has two subunit. Ribosome has two subunit. That is large subunit and a small subunit. Large subunit and small subunit. Okay. Small subunit, large subunit. They are present separately inside a shell, but at the time of protein synthesis, the main function of the ribosome is protein synthesis. At the time of protein synthesis, they bind together on the mRNA. It is a mRNA strand. It is a mRNA strand, and the ribosomes get attached with the mRNA at the time of protein synthesis. These are the large subunit and they are small subunits. For that, many ribosomes are attached with the mRNA at the time of protein synthesis. This structure is called polyribosome. Polyribosome or polysomes. Okay. Then type of ribosome. In case of the ribosome, we can classify the ribosome into two groups, that is prokaryotic ribosome and eukaryotic ribosomes. Prokaryotic ribosome is 70S and eukaryotic ribosome is 80S. So the prokaryotic ribosome also has two subunit, small and large. 70S has two subunit, that is 30S and 50S. 30S and 50S. Okay. Eukaryotic ribosome also has two subunit. Small subunit is 40S and the large subunit is 60S. Okay. Then the component of 30S subunit. Component of 30S subunit is the 16S rRNA, 16S rRNA plus 21 proteins. They are combinedly form the 30S subunit of prokaryotic ribosome. And 50S ribosome also contain 23S rRNA plus 5S rRNA plus 34 proteins. Okay, these are the constituent of the large subunit of a prokaryotic ribosomes. Then we come on to the eukaryotic ribosome. The 40S subunit means small subunit of the eukaryotic ribosome is made up of 18S rRNA, made up of 18S rRNA plus 33 proteins, plus 33 proteins. Okay, and 60S composed of 28S rRNA, 28S rRNA plus 5.8S rRNA plus 5S rRNA plus 5S rRNA plus 49 plus 49 proteins. These are the constituent of the eukaryotic ribosomes. What is rRNA? rRNA is the ribonucleic acid, means ribosomal ribonucleic acid, ribosomal RNA. Ribosomal RNA is the main constituent of the ribosomes. Okay. Then what is the S? What is S? S is the Vedberg unit. S is the Vedberg unit or sedimentation coefficient. It is the Vedberg unit. S means Vedberg unit or sedimentation coefficient. If we want to separate the different cellular part, okay, then we can use a process called centrifugation. Centrifuge is a machine in which we can separate the different cellular part depending on their atomic weight, and we can regulate the speed of the, we can control the speed, we can control the speed of the centrifuge, and in different speed, different part of the cell is propagated or centrifuged. In which speed we can get separated a part, that is the called Vedberg unit or sedimentation coefficient. Okay, so that is the structure of the ribosome. Then I come on to the microbodies. That is the last part of the cells, that is microbodies. What is microbodies? In cells, many small single membrane bounded structure also present. These are called microbodies. In the chart, I separated the microbodies into two parts, that is peroxisome and glyoxysome. Many others are also present, but these are the main microbodies which participate in metabolism. That is first one peroxisome. What is peroxisome? Peroxisome, it contains the peroxidase enzyme. It contains the peroxidase enzyme. For that it can synthesize hydrogen peroxide. Okay, for the presence of peroxidase enzyme, these microbodies also called peroxisome. Then another microbodies is glyoxysome. That is also a single membrane bounded small spherical structure which contains many enzymes for lipid metabolism. Lipid metabolism means breakdown process of the lipid. Okay, beta oxidation. So it it takes part into lipid metabolism, that is beta oxidation. The glycosome contains many enzymes which are participate in beta oxidation process. Okay, so the cell structure of cell is made now. Thank you.